Welcome everyone to this webinar, quite an international webinar. So good morning, good afternoon, or good evening to all of you who are attending. And yeah, just disclaimer. So this webinar is being recorded. And yeah, today we have quite some exciting talks, and we have with us, so in the schedule and the picture it shows way long, but unfortunately she's sick, and her slot will be filled in by Celia Fernandez-Sanz, who is a senior application scientist here at Singleron Biotechnologies. She will be talking for the first 10 minutes. Afterwards, we have Sophie Verkamplichter, who was a senior product marketing manager at Illumina. And it's quite early where she is. So thank you for being there at such an early time in the day. And our main speaker would be Dr. Mario Capasso, who is an associate professor at the Medical Genetics at the University of Naples Federico II, whose work mainly revolves around neuroblastoma, with quite an impressive CV and publications in Nature and NEJM. So thank you all for being here with us. Thank you for also the attendees for taking time out of your day, and we are hoping that you are going to come out with very good conclusions and something new learned.
So
I will hand it over now to Celia.
So yeah, I will be the first speaker, and thank you, Khalid, for the nice introduction. Let me share my slides with you. Okay, I hope you can see it properly. Let me present here. Let's go. Yeah. So today I'm going to talk and introduce Singleron Biotechnologies and a little bit about our products. So Singleron Biotechnologies is a biotech based all around the world. You can see here I am speaking right now from Cologne. Let me take my pointer here, where we have product development, sales, service, laboratory, and also tech support for Europe and part of the world. Also, we have also based, of course, in China for research and development, GMP manufacturing, a clinical laboratory, also in Nanjing and Suzhou. And also we have a base in Singapore and United States, Connecticut for North American sales and new technologies and development. So a little bit about single-cell sequencing. So you might be familiar with this picture of the smoothie and the fruit salad, and it's really to understand what is the dimension and what are the advantages of single-cell sequencing for your research. So when we do RNA bulk sequencing, it is like a smoothie where you cannot really distinguish which fruit it is there. It is a blueberry, it is a raspberry, a strawberry. Everything is mixed and confused. That's why we have a mix ingredients or a mix of cells that will give us a pooled results and of course an average profile of the entire sample. But we want, in certain cases, we want to go for a higher level of resolution where single cell sequencing gives us much more information. We will have in this case a fruit salad where we will be able to distinguish our cherry from the strawberry and the blackberry and the raspberry. So we start with individual cells, and we will be able to study the transcriptome of these individual cells individually, being able to get a precise profile of each cell types and sometimes even detect new cell types in our sample, which is not possible with bulk sequencing. So our technology, the core of our technology is the scope chip. The scope chip, you can see it here, is basically like a microscope slide the same size. And one of the nice things of this, many things that is nice, but I think one of the best things is that it's transparent, and you can always QC and observe your sample and how your workflow is starting and how it's going. So you can see this chip has 100,000 of microwells up to between 150,000 and 250,000, depends on the format you choose. We have all these microwells that are going to be to allow us to do the cell partitioning, that to separate our cells individually in each of these wells. Once our cells are introduced in our chip, and this can be do as easy as manual pipetting, the cells are going to drop in each of these wells. They are going to be compartmentalized in these wells by gravity, and then we are going to add our paramagnetic beads, which is with many probes on them, in order to later, when we lyse these cells and the cell coexists with one bead, we are going to be able to capture all the messenger RNAs from these cells. How does this work? The bead has millions of probes, and the structure of this probe is basically a read one for further sequencing and PCR amplification processes. It has a barcode, which is the same in each bead, but different among every bead, which is going to give us the single identity to our cells, and it's going to allow us to multiplex afterwards the transcript, which transcript comes from which cell. The UMI, these UMIs, or unique molecular identifier, are going to be all different within one bead, and it's going to allow us to get gene expression, protein, PCR bias, and also to distinguish doublets and background in our bioinformatic analysis. And of course, at the end, we have our poly-DT that is going to allow us to capture all by poly-A our messenger RNA. You can see here on the right how our microchip, thanks to the transparency of the microchip, can be observed under the microscope. And we have here all our empty wells that we can easily observe. These wells, in this case, are 40 microns diameter and 40 microns depth, so they easily accommodate one cell. And in each well, only one bead can be accommodated, so we are sure that only one bead goes in one well. And then when we load our cells, we can see here the cell distribution and then this nice cell partitioning of one cell on each well. After our cell partitioning and cell loading, we will proceed to introduce into the chip by simple pipetting or by automatization. I will also talk about that further in the slides. How the beads go into all of the wells, and in the cases where a well is occupied by a cell, we are going to see the coexistence of one bead with one cell. After this, we will proceed with cell lysis, and the probes in our beads are going to capture the messenger RNA for further library preparation. We have, of course, the number of wells, including the multiplexing option. We can customize the well size, and we have different number of wells density for different strategies and goals of your experiment. So we have in Singleron full solutions from tissue to tissue dissociation to single cell suspension until the end of your library preparation, compatible for Illumina sequencing and also for analysis. So you can see here we have products for the starting of our workflow, and also automatization for with machines for tissue dissociation and obtaining our single cell suspension. As I said before, we have also products for preparing your library, cDNA and library preparation. And also you can do this manually, and you can handle easily your chip manually, but there are options also for automatic chip loading and cell partition with our matrix machines, matrix and neomatrix. I will go further later more in detail about this. And of course, we have also our products for analysis and bioinformatic pipelines in Telescope and Synecosis. And finally, I will talk also a little bit about our in-house service option. So first, for the first part of the workflow, the stability of the tissue and the quality of the cell suspension is very important, and we have two solutions specific for this part of the workflow. One is the SL Life Tissue Preservation Buffer that can preserve your tissue for further analysis up to 72 hours at 4 degrees, and it has no chemical fixative and maintains your cell viability very high, up to 72 hours, and also our SL Life for tissue dissociation master mix, where we can get very high viabilities of your cell suspension, which is crucial for the success of your workflow. After that, we have also the automated tissue dissociation system with our Python Junior very mild, and where we can really get very fast, very nice cell suspensions ready to load in our chip. We have between 2 and 8 channels, and we can totally customize your programs with very starting with very little, up to 4 milligrams tissue inputs, and of course our tissue dissociation mixes. After you get with our solution the cell suspension, you can, as I said, manually load your chip, or you can use our automated cell processing systems with our matrix for 2 chips or 2 reactions, or NeoMatrix for up to 2 reactions in only 37 minutes. And then you can go for your reverse transcription, cDNA amplification, and library preparation after that. And this is the basic of the workflow, but of course in Singleron we have multi-omic options and also customizable options specific for your needs. We have, of course, as I said, the single cell transcriptome profiler, but also we can go for single nuclei and even yeast. We could go for transcriptome combined with VDJ profiling, CDR3 or full length TCR-PCR. We have also targeted sequence solutions for lung and blood cancer panels, chip, Epstein-Barr virus, and also customizable probes for your specific needs and goals. Protein surface glycosylation, we can detect the proteoglycans and the cell level sensitivity, and also a temporal resolution where you can study your transcriptome in the at that temporal time, at a temporal resolution and a temporal level, with a cell and gene level sensitivity where you can detect these newly synthesized transcripts. And finally, also what I wanted to I introduced before, our service. In our service, you can send us samples, both tissues or cryopreserved cells, and we can process all this for you up to the finalized workflow. We have already worked in more than 2,300 projects with more than 600 customers around the world. More than 320 million single-cell sequence so far, with different tissue samples focused on humans and mouse. But we have also worked with zebrafish, reptiles, C. elegans, all kind of tissues, and our success rate up to 94%, with the biopsy samples, surgical samples, and of course PBMCs and cryopreserved cells. So this is us so far from Singleron Biotechnologies. And also I wanted to add as a disclaimer, Dr. Mario Capasso, which is the main speaker today, did not receive any financial support from Singleron Biotechnologies. And I can introduce now Sophie. I think it's your turn.
I just wanted to say something I forgot to say. So if you have any questions, please use the Q&A button down there. You can write your questions and we will address them at the end. We will have like a Q&A session, and please, if you could write also to whom the question is addressed. Thanks. All right, yeah, Sophie, it's all yours.
Thank you so much for having me today. And let me just share my screen. Can you see my screen?
Yes.
Wonderful. So yeah, good morning, good afternoon, everybody. I'm Sophie with Illumina, and we have been collaborating quite extensively here with Singleron, and I'm happy to show you some data that have been generated together here. And so sequencing is really an important part of that single cell workflow, right? And I want to show you data that we've been jointly publishing with Singleron on our Illumina base space sequence hub. And so that was already presented before, but really sequencing NGS is a critical step of that single cell workflow. And the Singleron library, Celia was just presenting all the different products, but they are compatible with all the Illumina platform. However, single cell is a quite high intensity sequencing application. And because of that, we recommend the Illumina mid and high throughput sequencing platform. And so Illumina has a lot of sequencers that are here available, going from the left side to the small scale instrument, and on the right side here are highest throughput platform. And so when you think about your single cell experiment, you wonder what sequencer to choose, and that will really depend on how much data you need for your particular sample type, how many library you want to sequence, and of course, what sequencer is available. So if you have any question, you can always contact our tech support. We'll help you to guide you toward all these options. And so what I wanted to show today is we published recently some sequencing data set on our Illumina cloud that is called BaseSpace. And those data are free to access. And so together with Singleron, we have published sequencing data with the GEXScope library on the NextSeq 2000 and the NovaSeq 6000. And so we have those blog posts here that are available. And so here, there's the link. And so maybe you wonder how to get access to those data. So to do that, you will need to create a BaseSpace, Illumina BaseSpace account. This is free. And again, if you have any issue with that, you can always call our tech support. You need then to log into BaseSpace, and then you will access our demo data page with all the sequencing data that are publicly available that are accessible on that page. And so you will see there's a lot of sequencing run being published, but you can browse by using the single cell function. And so if you do that, you will see, for example, that NextSeq 2000 data with the single run library. And so you can click here on import your run or your project, and then you will just accept the run share. And so from that, you are able to go get those data. And so beside the sequencing run, we also just published recently an analysis part with our Dragon single cell analysis. So Dragon is the secondary analysis tool that we have at Illumina. And it's enabling analysis of the Singleron GEXScope library directly on the instrument for the NextSeq 2000. And so this is really pretty cool, especially maybe you work in a hospital type of setting, you cannot use the cloud. And so with that method, you can do directly the analysis on the instrument. And so for more detail, you can look at our blog post here, just published with our collaborator and colleague at Singleron. And that was it for me, really to give you a brief of introduction here. But I'm happy to answer any question. And so with that, we are going to have our main speaker today. And thank you again.
Yeah, thank you very much, Sophie. That was very helpful. And yeah, as you mentioned, this is the time I think everyone was waiting for. Our main speaker, Dr. Mario Capasso. Stage is yours.
Okay, can you hear me?
Yes.
Yes.
Okay. So, good morning to everyone. And in this my presentation, I will show you how the single cell transcriptomics have increased our knowledge about the tumor composition of neuroblastoma and also the molecular mechanisms that underline the chemoresistance in this tumor. So let's start to say what is neuroblastoma. Neuroblastoma is a pediatric cancer. This tumor can arise anywhere along the sympathetic nervous system, with the majority occurring in the adrenal medulla. And the median age is about, at diagnosis, is about 70 months. One of the most important features of this tumor is its remarkable clinical heterogeneity. And now, neuroblastoma holds a significant relevance in the field of pediatric oncology. In fact, as you can see in this figure, neuroblastoma accounts for 50% of all pediatric oncology deaths. And moreover, it is the most common malignancy diagnosed in the first year of life. So far, thanks to the genetic and clinical markers, we can stratify neuroblastoma patients in three main risk categories. As you can see here in this figure, we can have a high-risk neuroblastoma with very bad survival rate, is about 30%. But we also have low-risk neuroblastoma with very good survival rate that can be greater than 90%. And here I have listed the genetic markers and the clinical markers that we used to stratify the patients in these three categories. Another important feature of neuroblastoma is that this tumor is characterized by adrenergic and mesenchymal cell identities. In fact, two recent Nature Genetics papers, by using super-answer analysis of neuroblastoma cell lines, have demonstrated that there are neuroblastoma cells with adrenergic identity defined by a core regulatory circuitry with the module including these transcriptome factors. But we also have neuroblastoma cell line with the mesenchymal identity defined by these other transcriptome factors. Moreover, recently, another Nature Communication papers, by using a single cell RNA sequencing of 80 tumors, have demonstrated that adrenergic tumor cells can also present mesenchymal effusions, and this suggests the plasticity of these tumors. And this means that these tumors has the ability to switch from a mesenchymal to adrenergic identity, or vice versa. So, even though we have many genomic discoveries so far, and even though the patients receive this very aggressive treatment, including 3 consecutive steps and a combination of different chemotherapeutic drugs, the rate of patients that experience chemoresistance or relapse is still high. And we don't have too many therapeutic options in clinical practice. And this is probably due to the effect of the intratumor heterogeneity, ITH. You know that the tumor is not a monolithic structure of cells, but is composed of different cell identities. As you can see in this figure, here we have different subclones. And in these tumors, the cells can adapt in response to different selective environmental pressures with complex genetics and also with post-translational modification. And these give rise to phenomena of ITH. And during the treatment, as you can see here in this other figure, we have different subclones that can respond in different manner to treatment. And this can lead to the selection for a set of subclones, as you can see here, the red one or the green one, with existing or acquired therapeutic resistance. So it is clear that enhancing our understanding of ITH, there is a high impact on advancing cancer treatments. And now we know that the single-cell sequencing technology is a powerful tool to investigate the ITH, and also to understand the tumor origin and composition. And this was done in neuroblastoma. In fact, a recent Nature paper have demonstrated that by using single-cell RNA sequencing of different tumors, and also of adrenal tissue, this paper have demonstrated that neuroblastoma cells match, as you can see here. Here you have the different stage of fetal development, and here you have the different stage of neuroblastoma tumor. And these different stage of neuroblastoma tumors with a particular specific cellular composition match with a different phase of fetal development during the differentiation. And this paper have also demonstrated that neuroblastomas contain, this type of neuroblastoma contain late and mature neuroblasts, have a better prognosis than neuroblastoma with early neuroblasts. Another recent Nature paper have further reinforced the powerful of a single-cell RNA sequencing analysis in studying the ITH. In this paper, the authors have analyzed a large number of tumors. The data were single-cell RNA sequencing. This set of tumors cover more than 20 tumor types, and the aim of this project was to identify recurrent patterns of transcriptional ITH. And in this paper, these patterns were named metaprograms. The authors were able to find 41 metaprograms that occur in different cancer types. Moreover, they found 11 broader cancer hallmarks like cell cycle, cellular stress, mesenchymal lineage. And finally, they also found an association between these metaprograms and other clinical phenotypes or survival rate, and also an association with therapeutic resistance. In total, in this paper, we analyzed more than 2 million of cells, and the data are deposited in these databases. Other studies have suggested the characterization of tumor heterogeneity through the single-cell RNA sequencing, and they have demonstrated that this is a crucial technology to understand the molecular mechanism underlying drug resistance. So based on these observations, these papers, we developed this project in our lab to understand the neuroblastoma therapy resistance through single-cell biology. And our question is, what specific molecular pathways contribute to chemoresistance in neuroblastoma? Our hypothesis is the identifications of key genes and pathways associated with chemoresistance will provide targets for novel therapies. And of course, the methodology is the single-cell RNA sequencing of chemoresistant tumors. And here I show you our first results that was recently published. And here you see our study design. We performed in this study the single-cell RNA sequencing analysis of 2 high-risk neuroblastoma that were resistant to cisplatin and etoposide. We also analyzed their parental cell lines. And here we use the 10x technology, and we use the Surat package to perform the bioinformatic analysis. And here I show you the first results. As you can see, we obtain more than 40,000 cells for parental and resistant cisplatin cells, and almost 4,000 cells for parental and resistant etopocyte cells. And as you can see in this panel A and panel B, cisplatin and etopocyte-resistant cells show different transcriptomic profiles when compared with the parent neuroblastoma cell lines. Moreover, as you can see in this panel C, we found 3 clusters in parental and 4 clusters in cisplatin neuroblastoma cell lines. Instead, as you can see in panel D, we found 4 clusters in parental and 4 clusters also in etopocyte-resistant cells. And based on these results, we can conclude that neuroblastoma cell lines are transcriptionally heterogeneous, and we can also say that drug resistance can induce the development of cell subpopulations, and these findings emphasize the phenomenon of ITH in neuroblastoma resistant cells. We also observed that chemoresistance can affect the neuroblastoma identity. In fact, as you can see in this plot, in these different plots, all cisplatin-resistant clusters are enriched in adrenergic genes. Instead, we found etopocyte-resistant clusters equally enriched in mesenchymal and adrenergic genes. And these results highlight this plasticity of etopocyte-resistant cells. And so, as I told you before, their ability to switch from mesenchymal to adrenergic identity. We also perform a gene ontology analysis of each cluster found in parental cells and drug-resistant cells. As you can see in this figure, we found that in parental cells, there is enrichment on gene pathways involved in regulation of DNA, RNA, metabolic process, and also the development of nervous system and organ in general. We also, instead, in drug-resistant cells, we found gene pathways involved in cell cycle regulation, DNA repair, and protein post-transductional modification. So, based on these results, we can conclude that the drug treatment can induce acquisition of DNA repair and post-translational modification pathways that are relevant to induce drug resistance in neuroblastoma cells. We also demonstrate in this paper that the ITH of neuroblastoma cell lines can predict neuroblastoma outcome. In fact, we perform a deconvolution analysis of transcriptomes of almost 400 bulk neuroblastoma tumors, using the cell population clusters that we obtain by single-cell RNA sequencing in neuroblastoma cell lines. And as you can see in these 2 plots, we found that the ITH is comparable to that observed in parental drug-resistant cell line populations. Moreover, we also correlate these clusters with the clinical markers and genetic markers of neuroblastoma, like risk categories, stage tumor, MICAN status, and age at diagnosis. And as you can see in this plot, we found diverse clusters, while belonging to the same cell lines, are associated with different positive or negative clinical factors. In fact, as you can see, for instance, in this parental cell line, the cluster 0 was associated with high risk neuroblastoma, but the cluster 5 in the same cell line was associated with low risk neuroblastoma. Also, in these cisplatin-resistant neuroblastoma cell lines, we found the cluster 2 associated with high-risk neuroblastoma, but the cluster 3 is associated with low-risk neuroblastoma. And we also observe that these two relevant clusters, the cell cycle cluster and the metabolic biological process, found in cisplatin etoposide resistant cell lines, are strongly associated with all high-risk tumor phenotypes, including also the poor survival, as you can see here. And these data suggest that these two clusters are relevant, are very aggressive cluster in neuroblastoma. Moreover, these two clusters can be also potential gene signatures to identify non-responder neuroblastoma patients. So in conclusion, for this is my first part of my talk, we can say that we have demonstrated that neuroblastoma cell lines can have distinct tumor subpopulations, and this can determine the ITH of drug-resistant neuroblastoma cells. Moreover, we demonstrate that drug resistance affects neuroblastoma cell identity, and in fact, neuroblastoma-resistant tumors can show both adrenergic and mesenchymal identity. And this again support the concept of plasticity of neuroblastoma tumors. And in the end, we show that the ITH of neuroblastoma cell lines reflect the cellular composition of neuroblastoma tumors. And we also demonstrated that this ITH can predict the clinical outcome of neuroblastoma patients. So to conclude my presentation, I would say that single-cell RNA sequencing is an available tool for unraveling intratumor heterogeneity that can contribute chemoresistance. Moreover, this approach, this technology can track the clonal evolution of tumors over time. And this is very important to help researchers to understand how certain clones of cells, which may be initially sensitive to treatment, or evolve to become resistant under the selective pressure or therapy. Another point is that this technology is very important to comprehend the molecular mechanism responsible for drug resistance in the treatment of tumors. And this information is very important to develop target therapies and to overcome resistance mechanisms. In the end, I think that we need technological advancements to analyze low-quality samples, like our samples, and also to assessing the DNA alterations that so far are little investigated. And before I conclude, I just want to thank my collaborators, especially Ferdinando Bonfiglio and Marianna Vitabile, that did most of the results of experiments that I show you today. And I want to also thank all other participants to this project and our grants. And of course, thank you for your attention, and I will be happy to respond to your questions.
Thank you. Thank you very much. That was very, very interesting. And I worked in the hematology department before, and I know of people working on neuroblastoma. And the heterogeneity is, of course, a big problem in cancer research. So for everyone, you can, of course, direct your questions via the chat. So there is this Q&A button on the bottom. So if you have any questions to any of our speakers today, please type them in, and we would direct them. I will read them out, and we will have the answers. In the meantime, I can maybe ask some questions from the audience, but also from me. So for the cell lines, were these primary cell lines taken from patients?
Yes. They are established cell line that we use usually in neuroblastoma. And this was unexpectedly results for us. Because usually established cell lines are homogeneous, as you know. But we another research group have demonstrated that also in established neuroblastoma cell line we can identify this intratumor heterogeneity. We can identify this subgroup of cells.
Okay. And for the clusters, you talked about the ones that were important that you discovered. So did they have a certain phenotype, so certain cell annotation?
Can you repeat, please?
The clusters that you identified at the time in those primary in the cell lines. So were they annotated differently? Did they have a certain?
Yes, of course. Yeah, the clusters that we found in this part of our results was very interesting because we found in parental cells the clusters that belong to the pathway related to neuron development that is in line with the origin of neuroblastoma. In contrast, in the resistance cell line, we found pathway involved in the drug-resistant mechanism like the cell cycle, like the DNA repair. So these are very important because first of all, we confirm the validity of our method, and second, we also know now that there are some cluster of cells in resistant tumors that can be targeted in a targeted way.
Okay. And the plasticity that you mentioned, so is it that the cells are shifting, you think, from one phenotype that's adrenergic, or is it that they are dying? One of them is more sensitive and it's dying and it's shifting the phenotype?
You mean in a neuroblastoma cell lines or in tumor in general?
In tumor in general, but what you find, because you mentioned the plasticity that you found.
Yes, we found in neuroblastoma cell lines that in chemoresistant cell lines, we found both adrenergic and mesenchymal lineage. And this is a very, usually I was expected to find more mesenchymal subpopulation because we know by literature that mesenchymal lineage is more resistant to drug. But in this case, and we found both type of lineage. But now there is a new paper that have found similar results. So this further reinforced the concept of plasticity and also reinforced the concept that also adrenergic neuroblastoma can be a subpopulation of cells resistant to drug.
Okay. And for the single nucleus and the single cell, so have you compared them, the results that you get from both?
No, I never done this. But as you know, in our lab, in this project, our samples are very rare tissues. We only have fresh frozen tissues that are sometimes also old tissues. And so I would say that I'm pretty satisfied from this single nuclei analysis because of course we were not able to analyze all samples that we have sent to the Singleron, but most of these were successful analyzed.
Okay, this is good to hear. So I will read out one question also that came. I think this is for Xenia. So what type of sample can be used with this chip? I guess the chip that you presented. So is it fresh tissue, fresh frozen, cultured cells?
Yeah, so the chip is compatible with any cell suspension that you can get. So as I'm going to share here this slide, I don't know if we can see it.
Yeah.
So we can process this press sample or also fast frozen for single nuclei as Dr. Capasso just explained, we can do cryopreserved cells and any kind of tissue that you can, of course, preserve in a preservation buffer if you need to process it later. As long as you get a nice and good and healthy cell suspension, these cells can go into the chip. And initially, yeah, you can see here we have processed lung, liver, ovarian, endometrium, bladder, brain, blood vessels, even bone or breast, any kind of biopsies, cancer, skin, anything, basically anything that fits. So one of the limitations of the chip is the cell size, but we have also specific options and for big cells, but up to 30 micron cells, they can be introduced into the chip. And also the same way and the same kind of workflow, in case there is a flash frozen tissue, we can isolate nuclei and also introduce them into the chip. So basically everything that fits and it's healthy can go there.
Okay, so I hope this answer was satisfactory to whomever asked the question. A question for Sophie. So for this Dragon platform, so you said the analysis is possible also for Singleron data. Does that mean that you can go from the FASTQ files down to the gene count matrix?
I might have to take that one offline for the analysis. Yeah. All
right. Okay. Another question for Dr. Capasso. So just for, you mentioned one of the ways to stratify if it's a good or bad responsive is the diploidy. Did I understand correctly?
Sorry?
One of the ways to stratify the response or whether it's a good or bad, like is it high survival, low survival, was the diploidy? If they are diploid?
No, the markers that we use to stratify patients,
yes.
We using different clinical markers like tumor stage, because in neuroblastoma we have 4 tumor stages. The stage 4 is the most aggressive tumor, and we also use the risk category that are established by guideline of neuroblastoma, and we also utilize the MICAN amplification, because it's the most important genomic marker to define the risk and to stratify the patients according to risk. And we found that this different cluster in that we have obtained by the convolution of tumor bulk are strongly associated with these markers. So this means that the cluster that we found in neuroblastoma cell lines can be reflect the heterogeneity of tumors, and these clusters can be associated not only some clusters associated with bad survival or bad clinical markers, but other clusters are associated with good survival and good clinical markers. And these results reinforce the concept of heterogeneity, intratumor heterogeneity in neuroblastoma.
Okay. All right. Do we have any other questions? Last chance. All right. If there are no more questions, I would like to thank all our speakers today. This was really enlightening, I think, for me, and I guess for everyone attending. Yeah, really, thanks a lot for all your time, and yeah, see you, I guess.
Bye-bye.
All right.
Bye.
Bye.